Our next speaker is Uttam Bhattarai. He is a PhD student at Louisiana State University in Baton Rouge, Louisiana, and he is working with molecular genetics of rice resistance to drought.
Hi, good morning, everybody. I'm Uttam Bhattarai from Louisiana State University, and lots of talks going on today about the drought, and I'm just continuing the same talk. It's about drought, and I'm trying to identify some of the QTLs that are response to the drought resistance in rice. And this is my rice field that I had grown like 2 years before, and we stopped irrigation in this field for almost 10 days, and the field looks like this. And this type of conditions are severely seen in the rice growing areas in Asia, where the cultivation is solely dependent upon the rain, where there is no irrigation facilities. And the drought tolerance in rice is targeted to those type of situations where there is no rain. Where there is no irrigation facilities and rain is there's no prediction of the rain. We all know that rice is hydrophyte. It needs a lot of water. It needs a lot of standing water to grow all entire its growing period. And the rice in like 45% of the rice grown all over the world is rain-fed. It's dependent only upon the rain. They do not have any irrigation facilities, and that's mostly in this southeastern part of Asia. And in Sub-Saharan Africa, it's almost 80% of the land is dependent upon the rain. And the losses due to drought is very severe. You can see some of these drought years in India. The yield is drastically reduced, and this experiment also shows that as the number of the days without water goes on increasing, the yield drastically goes on decreasing. And the production loss of rice is almost like 2/3 of the loss may occur due to drought. And drought may occur at any time of the rice growth. And the drought that occurs at the early stage, that's called vegetative drought, and the drought that occurs during the growth stage, any other growth stage of the plant, that is intermittent drought, and the terminal drought that occurs at the flowering stage. And this is very severe, as Dr. Prasad already mentioned, that the flowering stage drought is very severe. And although we have some of the strategies, that conventional strategies we can use to just escape those drought conditions, but since drought cannot be predicted, so we need to get ready for any such circumstances to cope with this situation. So we tried to do some mapping works to find the candidate genes and the QTLs involving the drought tolerance in rice. Cocodri is a drought susceptible variety. That's Louisiana varieties. It's released from Louisiana. And N22 is a drought tolerant varieties that's popular all over the world. And we try to focus on the underground traits, that's roots. So we just try to use these long pots, that's 75 centimeter long pots. And we set experiment in our tunnel like this, and we try to we fill them with soil and sand so that it will be easier for the soil to take out while we are measuring the root. And we just grow them regularly for almost 5 weeks, and after 5 weeks we impose the drought during the vegetative growth stage, where there is a maximum growth stage of vegetative growth stage of the plant. So there's lots of activities at that time. And we measure some of the root and the shoot traits to evaluate. We just did genotyping. For genotyping, we used this GBS technology that the Cornell University, we just sequenced our data from Cornell University. We generated almost we have the F7 population of those crosses, and there are almost 190 lines we are using for the genotyping of genotyping. And sequencing was done at Cornell University, and we used tassel pipeline to identify the SNPs for those genotyping works. And we manually also removed a lot of SNPs that we feel that like they are not good. So we just removed almost all the SNPs that are more than 10% of the missing data, and all the monomorphic SNPs are also removed. And heterozygous SNPs are just encoded as the missing SNPs. And finally, we ended up with almost 47 to 4,800 SNPs, and that was used for mapping. Using QTL mapping was done using interval mapping and composite interval mapping methods in ICIM. This is just the phenotypic visualization of the traits that we have measured. We measured that the root length goes on increasing while we impose the drought. This is the percentage change of the traits while in a stress and a non-stressed condition, and the root length goes on increasing. That's a very well accepted thing, that well accepted research that found that as the drought increases, the root goes deeper and deeper and deeper. And the increase in root length was more pronounced in N22, that is the donor for the drought tolerant cultivars. And other traits mostly goes in decreasing, except for number of TLS that goes in increasing. And the root shoot ratio also goes in increasing because of the root length or maybe root mass. This is just a quick view of the distribution graphs of various traits. And they have like cocodria and N22, we have the populations, and the real mean is lying in between them or in one side of the population of the parent, and we have a lot of transcriptive segregants in between them. This is the SNPs summary of our SNPs, linkage map that we have drawn with. We ended with almost 47, 48 SNPs, and yeah, the total genetic length was almost 1700 centimorgan, and the total physical distance was 365 MB in our genetic map. This is the linkage map that we have drawn from the ICIM mapping, and there are some of the hot spots where we find a lot of QTLs, and chromosome 1 has a lot of drought-tolerant QTLs. Many people have found a lot of QTLs in those regions also, and we also found a lot of QTLs in those regions. These are the list of the QTLs that we have identified. A total of 33 QTLs we identified for 8 agronomic traits. And root length that we consider the most important for drought, we have one QTL identified at chromosome number 12, and it has not much phenotypic effect, but 5% of phenotypic effect. Besides this, we have a lot of other QTLs that have very good phenotypic variants, like for shoot length in chromosome, I have named this as a chromosome 1 at 37 MB region, chromosome 1 at 38 MB region. So these are almost a lot of phenotypic variants, 16, 19, 17% of phenotypic variants for shoot length. Shoot length is drastically reduced while during the drought conditions, and we found this type of QTLs that are very useful for drought tolerance too. This is the continuation of the QTLs that we have identified, and the major QTLs that I have identified for these are like having the 6 to 11% of the phenotypic variance. That's quite good, and that is for root mass and shoot mass and root shoot ratio. Besides this, we have found some of the common QTLs in between for those traits, like shoot length QTL in chromosome 1 is the same as the root mass QTL in chromosome 1. Therefore, we can just conclude that the same QTLs may affect the root and the shoot things. So maybe it's a set of genes that are like interacting with each other to govern the root and the shoot traits. Besides this, we also try to find the genes in between those QTL regions. We have almost 1,800 genes that we found in between those QTL regions, and we try to annotate those genes functionally using the Panther classification system. And that identified almost 80 genes that have the functional annotation, and that's the number of genes for different traits are quite different. And these are also classified based on the 4 major functional classification systems. And finally, we ended up with some of the very good genes that have previously also been said, like the important genes for stress response. Like this gene family, putative protein, Brevis radix-like protein, this protein is very crucial for the proliferation of the root, and we found this protein in our gene. We found the gene interacting with this type of proteins, and we listed. I tried to list some of the like good genes that have previously mentioned in a lot of literatures that are responsible. So we ended up with this type of candidate genes, and these genes can further be need to be validated to make sure that these genes are involved in drought response. And that's all. Thank you. Thank you for your question. Any questions?
Thank you. Any questions?